And here he is, Pateke, also known as the brown teal. In 1882, Walter Buller said that he found these birds all over New Zealand. They were apparently in every inland lake and in all the freshwater streams that ran into them. They were also often seen foraging in forests, quite some distance away from the water. Fossil evidence would suggest that they've been around for about 10,000 years and were widespread around New Zealand and really abundant. In fact, so abundant that some estimates suggest their numbers would have been into the millions. But unfortunately, like so many other New Zealand species, the numbers have seen a sharp decline. And now the population of the brown teal is thought to be under 1,000, with populations on Great Barrier Island, the east coast of Northland, and 6 offshore islands, with 3 of these offshore island populations resulting from translocations of animals by scientists in order to help conserve the species. As humans arrived in New Zealand, that's when the problems for Pateke really started. Unfortunately for Pateke, they seem to make a pretty tasty meal, and this is often the reason that they were hunted in really great numbers. And if that wasn't bad enough, the dogs, cats, rats and stoats were also partial to Pateke. At the same time as all this was going on, humans also drained 90% of New Zealand's wetlands and removed up to 70% of New Zealand's native forest. So it's not surprising that now Pateke, like a lot of New Zealand species, is facing a real threat of extinction. And this is recognised by the International Union for Conservation of Nature and Natural Resources, that lists Pateke as endangered. But at least all is not lost. Pateke still survive. And New Zealand's a world leader when it comes to raising the numbers of threatened species. So let's have a look at what's being done to help them, and then let's find out what you can do to get involved. So one big part of the recovery plan for Pateke involves a captive breeding program. And this involves breeding the Pateke in captive, but then reintroducing the chicks into the wild, and then obviously raising the numbers of the number of Pateke in the wild. I'm sure you've seen lots of other examples of this, for examples with kiwi and things like that. You maybe even seen them in Rotorua. But it's an expensive and a time-consuming exercise. So we have to do everything possible to make sure that that time and that money is spent well and that the breeding program is successful. And that's where Landcare Research and your good self actually come into this. So as we've already said, the numbers of these birds has gone down from numbers into the millions possibly, down to under 1,000 in probably the last 1,000 years. And this is a really good example of what we call a genetic bottleneck in a population. And this is where you've had a once really large population with a high level of genetic diversity, and then that population quite suddenly becomes quite small, and with a small population is a much lower level of genetic diversity. And small populations like this, with low levels of genetic diversity, are really prone to the effects of what we call genetic drift. And this causes even more problems for these small populations, because with a lower amount of genetic variation comes less opportunity for this organism to adapt to its environment, which again then increases its chance to become extinct. So what we need to do is choose birds for the captive breeding program that have got quite a high amount of genetic diversity, so they produce chicks that have also got quite a high level of genetic diversity. So not only can we increase the population size of the Pateke, but we can also maintain the diversity that it's already got. So what we need to do is figure out which chicks have got the best amount of genetic diversity so that we can choose the right individuals for the breeding program. Okay, so let's start by recapping on what exactly a DNA profile actually is. Well, to explain it, I want to actually look at another type of profile. Let's say a description of a person's profile in a physical way. So when you describe someone, you might describe a number of their characteristics to actually allow someone to identify who you're talking about. Characteristics such as hair colour, eye colour, height, size, weight, race. These are all properties of a person's physical appearance that we can use to describe what they're actually like. Well, a DNA profile is pretty much the same thing, except what you're actually doing is you're describing different parts of their DNA. On the screen right now is a diagram that shows all the different parts of a human genome that we use to create a genetic profile. So all those little labels there represent part of the DNA that we describe when we're actually creating that DNA profile. So, for example, we might describe a person's DNA in terms of what is the DNA like at where it says TPOX on chromosome number 2. Or we might describe that person's DNA in terms of what it's like at TH01 on chromosome number 11. Now, each of these points on the diagram on the screen has been chosen because it actually contains what we call a microsatellite. And microsatellites have got other names as well, sometimes known as short tandem repeats, variable number tandem repeats, all kinds of things like that. But basically, it's a region of the DNA where there's been a repeat in the genetic code. On your screen right now, you'll see an example of a microsatellite. This microsatellite is what we call a dinucleotide repeat. And you can see that we've got some base pairs or some bases on the screen there, but you'll see that GA is repeated 6 times. Now, what we find within a species is that they all have certain areas of the genetic or the genome where we have these areas where there is a repeat of our bases or a microsatellite. So if we think back to the picture we were just looking at a second ago, we maybe just have a quick look there at the area of the genome called TPOX, and we'd notice that that's an area where there's a repeat. But the important thing is that not every individual will have the same number of repeats for that particular microsatellite. So some people might have 6 repeats, some people might have 8, some people might have 10, some people might have 7, and so on. And it's the same with all species. So by looking at a large number of these microsatellites, say for example in the case of humans here, 13 different areas of the genome, 13 different microsatellites, we can then put a picture together. So how many repeats was there, say for example, at the TPOX area, and how many was at the D3S1358, and so on. And basically you then end up with 13 different signatures, if you like, in the genome. So there may be 6 repeats at one, 8 repeats at the other, and the more different places you actually look in the genome, the bigger or the more detailed the picture gets of this particular individual's DNA profile. Now, this particular piece of work that we're looking at there is really important for the work we're going to be doing with the Pateke, because the population is really small now, less than 1,000. That's quite a small population, especially compared to the fact that it was once in the millions. And with that small population, we're expecting to see quite a low level of genetic diversity. So you'd expect a lot of individuals to have pretty much the same DNA profile, which means they're genetically very similar to one another. So what we're going to do is use the DNA profile to find out, okay, well, which ones have got the most diverse profile. So rather than just have the same as everyone else, which ones have got the most diverse, so the most, if you like, unique DNA profile that contain the most different types of microsatellite repeats. And if they've got a really different DNA profile in terms of the microsatellite regions that we're looking at, then we can infer from that that they've also got quite a diverse genetic makeup as well. So they've got quite a high level of diversity in relation to other members of their population. And remember, it's those individuals we really want to use for the breeding program. So we want the individuals to be in the breeding program to have quite a high level of diversity so that they are likely to pass on a wide range of genes to future individuals in the population. So that's all well and good. What we need to do now then is we need to let you know how we can actually figure out which individuals have got which number of repeats in the microsatellites. And to do that, we're going to use a few biotechnological techniques that you'll be familiar with, hopefully. And if you're not, then you can do some work with this, or you can actually teach you a little bit more about it. And we're going to talk about using polymerase chain reaction, or PCR, and maybe also talk a little bit about gel electrophoresis. So let's start with polymerase chain reaction. So you should all know by now that PCR is a reaction that we can use to amplify or copy a specific section of DNA so that we've got pretty much 1 billion copies of it in a short period of time of about an hour and a half. So it's a technique we use to copy one area of the genome many times. Just see if you can remember what you actually need to put into the PCR vial, the actual little thing that you put in the machine. What different things do you actually put in there? Maybe pause me for a second if you need to have a time to have a think about it. Okay then, so here we go. First of all, we need the DNA sample that we've actually got from the individual we wanted to profile. Then we also want some DNA polymerase, but remember it's Taq polymerase that we actually use in PCR, and the unique thing about that is it's actually thermostable, so it can cope with really high temperatures, like we're going to actually put it through as part of PCR. Also we need some nucleotide bases, so that's the A, T, C's and G's, and all the sugars and phosphates that are attached to those. And then finally we need 2 primers for each of the microsatellites that we're actually wanting to amplify. So for each specific sequence we want to actually copy, we need a primer for either side of it. And those primers will be what allows the Taq polymerase or the DNA polymerase to actually bind onto the DNA and then copy that specific sequence. So if you need a little bit more help with the PCR thing, there's a really cool website, it's just on the screen right now, that if you go to that, it's called, what it's called a virtual lab. And the virtual lab will actually talk you through the whole process of PCR, what you do in the lab to make it work, and then if you could actually see all the little DNA molecules, what actually would be happening to them. So it's quite a neat little resource, even just to use as a refresher. Okay, so the bit that we're most interested in is the primers and what they do in PCR. And on your screen now is a diagram that will help me explain a little bit more about what primers do. So the thick set lines there, the black lines that are going horizontally across the screen, with one of the 3 on the left hand side and one of the 3 at the right hand side, they represent the DNA. And at this point in PCR, the DNA has been denatured, so heat's been applied and the hydrogen bonds have been broken and the 2 strands of DNA have been broken apart. And it's at this point here that the primers actually attach to the DNA strands. And you'll notice that they attach either side of the target region depending on which strand they're actually going to fit onto. And they do this so that the DNA polymerase or the Taq polymerase can bind on top of the primer and then start adding new bases or new nucleotide bases onto the 3 end of the primer to start synthesizing the new piece of DNA and amplifying the target region or the target area that we want to actually copy. Again, if you're finding this difficult, go and watch the virtual lab that I showed you earlier on. Okay, so this diagram is a little bit more specific this time. Same sort of stuff, but just with a particular example and a particular sequence of DNA that you can actually look at. Once again, you can see that this double-stranded piece of DNA has been denatured, so it's actually split apart. And the primers have actually attached to each of the strands. And you'll notice that they're attached either side of the microsatellite region, which is a region where you see the repeats. So on the top strand there you can see AC, 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 which is an AC repeat, which is repeated 8 times there. And on the bottom, on the other strand, you can see it's a TG repeat, TG8. So obviously these primers are designed to actually copy that particular microsatellite region. And if PCR was done from a sample of DNA from a particular individual, PCR would make a specific copy of that microsatellite. So this individual's got AC that's been repeated, say 8 times. Another individual might have it repeated 10 or 12 times. And we could measure the number of repeats that an individual actually possessed in their DNA by at a later stage doing a gel electrophoresis. So we can actually separate the pieces of DNA according to size and compare it against known size fragments of DNA so we can actually figure out the length of the microsatellite for a specific individual. But more likely we're probably just likely to put all this DNA from the different PCRs into a gel and actually create an image of the DNA profile. Much the same as the one you can see on your screen there. With each the vertical lines of bars there representing a different individual. The bars closest to the bottom of the chart there indicating fragments of DNA that have quite a big length comparison to the bars above them, which are fragments of smaller lengths. But I'm sure if you're interested in learning more about gel electrophoresis and how that works, you could follow the link on the screen right now, which is another virtual lab similar to the PCR one I talked about earlier on, and you could go back and have another look at that. Okay, so looking back at the screen now, we've got the diagram we were looking at just a second ago, and this is really sort of going to allow me to explain exactly what you're going to be doing in this project should you get involved in it. So you're really going to be looking at identifying the microsatellites and then designing the primers that allows PCR to actually make copies of those microsatellites, which will ultimately allow us to decide which are the most genetically diverse individuals, so we can ensure that we continue to maintain the genetic diversity of the population over current generations and in the future, and prevent the genetic diversity from reducing due to the active genetic drift. So we're going to use the DNA sequences that have been sent to us by Landcare Research, And we're going to use some free to download pieces of software, and then we're going to use Microsoft Excel to do this. So those of you who actually do get involved in this will actually actively be part of this research. You'll actually be designing the primers that the scientists over at Landcare Research are actually using in their research to identify the DNA profiles of the potential individuals that are going to be used for this captive breeding program. So you really are involved. You're not just sort of doing something as a practice. This is actual real science that you're actually participating in. But I'm going to end this podcast here now, as I've told you exactly what it's all about. And if you're interested in moving on with this, then you're going to need to watch the next podcast, which is all about how to actually do this, how to actually design these primers, how to identify where the microsatellites are, so that we can actually start genotyping or DNA profiling these individual birds and figuring out what's what. So thanks again for listening and watching. And I'm looking forward to working with you to actually do this work and identify the microsatellites and be the first people in the world to create primers for this particular species of animal, which obviously by the sound of it needs a little bit of help. So once again, take care, speak to you on the wiki, and as always, keep it real.